Hi everyone, welcome to our Thursday webinar series. My name is Lourdes. I'm glad that you are with us here today. Today we have Caitlin Van Lith, who will be speaking about the global genetic study of inherited risk of cholangiocarcinoma. Caitlin is a sixth-year MD PhD student at Mayo Clinic, performing her thesis work under Dr. Lewis Roberts. Her projects focus on clinical and genetic risk of liver and bile duct cancers, as well as global genetic equity in scientific studies, and we're glad to have her here. As a reminder, please put any questions you might have into the chat or Q&A, and please make sure that your questions are general in nature on this topic. Welcome, Caitlin.
Thank you. Are you seeing presenter view or?
I see the presenter view.
Okay, now you see.
Yep, perfect.
Perfect. All right. Thank you everyone for joining today. As Lourdes said, my name is Caitlin Van Lith. I'm an MD PhD student. So an MD PhD programs, I did the first 2 years of medical school. I'm now getting my PhD in clinical and genetic risk factors of cholangiocarcinoma and hepatocellular carcinoma, which is liver cancer. And then I'll go back, finish med school, and then go to residency. So I'm in training, but I've been working in this space for the past several years. And Dr. Lewis Roberts, my PI, has built his career in this area. So today we're going to review cholangiocarcinoma, and then we're going to delve into genetic sequencing and the genetics of cholangiocarcinoma, and then talk about this specific project that we're collaborating with CCF on. So cholangiocarcinoma is a cancer of the bile ducts. It can present in different areas of the biliary tree or liver, and we call it different things based on where it is. It can be intrahepatic, it can be extrahepatic, which is either perihilar or distal. There are some really well-known risk factors for cholangiocarcinoma, such as colorectal cysts, biliary tract stones, primary sclerosing cholangitis, and other autoimmune conditions, and liver fluke infection, which is more prevalent in Southeast Asia. But a lot of cases have no known cause. So there's a really important emphasis here in early diagnosis. And in early diagnosis, prevention and screening are key. But then the question is who should we be screening? And so this is where genetic studies can really come in, is helping us identify screening populations. The incidence of cholangiocarcinoma, so incidence is how many people in the population have it, really varies globally. In most places in the world, including the United States, it's a rare cancer. In some parts of Southeast Asia, where that liver fluke that I mentioned is prevalent, it's not considered a rare cancer. But the thing that I want you to take away from this figure is how many countries are in the grayed out zone. So we're seeing data from the United States, Canada, Europe, Southeast Asia, and Australia, but a vast majority of the world in this diagram is grayed out. And we're going to come back to that in talking about the specific project that I'm working on. Going back to the risk factors for cholangiocarcinoma, we called out some of these. We have kind of a metabolic cluster. We have, some environmental and infectious type things, the liver fluke, viral hepatitis. We have autoimmune diseases like primary sclerosing cholangitis, ulcerative colitis, some environmental toxins that might play a role, and then genetics, which can really influence a lot of these different groups. And kind of the common thread in all of these risk factors is chronic inflammation. And so in our study, we're really focusing on how genetics is influencing cholangiocarcinoma development. And this could be through other risk factors or directly into cholangiocarcinoma development. So now we're going to do a little primer on genetic sequencing so that you have a good background in understanding what the study that we're doing is. So as a review from high school biology or college, if you took it there, our DNA, we have 23 pairs of chromosomes. And in the DNA, there are different segments that make up our genes. And not all of your DNA makes up genes. Only about 1 to 2% of it are these genes. And the rest is what we call non-coding DNA. But within all of DNA, it's made up of these 4 different letters, A, C, G, and T. And that's what codes for everything that your body does. It's a pretty cool system. But there are essentially mistakes that can be made. And so a very common one of these is where a single one of those letters turns into something else. And this is called a single nucleotide polymorphism. So single nucleotide polymorphisms, or SNPs, it's the most common type of genetic variant. A common word for this is also mutation. The field has tried to move away from mutation because they're not all bad. Mutation kind of denotes that it's a bad thing. Not all variants are bad, but some can be, certainly. And in the vein of not all of them being bad, this occurs super normally. So about 1 in 1,000 of these letters has a single nucleotide polymorphism, which means that any single individual has 4 to 5 million SNPs in their genome. And obviously, all of us don't have 4 to 5 million different diseases. So these are almost always non-disease causing. But variants within important parts of genes and even outside of those genes can affect the protein's function. And proteins need to function a certain way in order for our body to function properly. And so one of the things that an affected protein could do is predispose someone to cancer. So there are different ways that we can look at the genome to look for these SNPs. One of the earliest and most basic ways that we can do it is what's called a candidate gene panel. So that's when we sit here and say, okay, we think that genes XYZ are involved in cholangiocarcinoma development. So we've identified these relevant genes, and then we say we're only going to sequence genes XYZ. So this is most clinically used. If any of you have gotten a clinical genetic panel, it was likely this candidate gene panel. Some of you may have gone and had whole exome or whole genome sequencing, but this is by far the most common type of clinical sequencing. It is the most hypothesis driven. So we identify ahead of time what we think is relevant, and then that's all that we test for. And a strength of this is that studies don't require as large of numbers because you're not getting as much data. So these studies can be performed in smaller groups of patients as opposed to agnostic sequencing. So in agnostic sequencing, we're not pre-identifying relevant genes. There are a couple of types of agnostic sequencing, but one is whole exome sequencing. That's when you're only coding genes in those protein coding regions, which is about 1 to 2% of the genome. And you can see in this diagram here, that would just be sequencing these dark green areas, these exons. So 98 to 99% of the genome is this light green, but the dark green is what codes for proteins. And so theoretically, those are the most valuable to have information on. So in whole exome sequencing, we're sequencing those dark green areas. And then there's whole genome sequencing, where we're sequencing both dark green and light green. And there's a variety of reasons why you would do each of these different things. As I said, candidate gene panels gives you the least amount of information, but it's also the cheapest and it's the easiest to interpret. Whole exome sequencing is in the middle, but closer to whole genome sequencing when it comes to interpretation, cost, and information given. And so the higher level of sequencing that you do, the more complicated interpretation becomes, and the more that you need people who are PhDs in this area, in bioinformatics specifically, It's not something that I can interpret on my own. And it's not something that most physicians can interpret on their own, but it does give a ton of information. And one of the ways that you can do whole genome sequencing is through a study type called genome-wide association studies, so GWAS. And this is something I'm going to refer to throughout this presentation because that's really what the project that I'm working on is about, is a GWAS study. So in a genome-wide association study, and in our specific genome-wide association study, we're sequencing germline DNA. And so cancer patients have two different types of DNA. They have germline DNA, which is what you're born with. That's what's circulating in your blood. Then there's also tumor DNA. And so since cancers have mutations that cause that tumor, those are going to be slightly different. They're almost the same, but those mutations that cause the cancer are going to be different. And so if you've been told that your tumor has an IDH mutation, for example, that would be your tumor DNA likely, as opposed to your germline DNA. So we're looking at inherited risk. So looking specifically at this germline DNA, we're going to be sequencing it and looking for those single nucleotide polymorphisms, or where there are just those really small changes in DNA. And we're going to be sequencing large numbers of both cholangiocarcinoma patients and those without cholangiocarcinoma diagnosis. And you can see in this diagram here, essentially what we're looking for is if there are differences in how frequently we see a SNP, a single nucleotide polymorphism, between the two populations. And so in this red group of patients without cholangiocarcinoma, we're only seeing 1 in 3. But in the group with cholangiocarcinoma, we're seeing 2 in 3. So that's a very simplified version of what we're looking for in GWAS studies. And this allows us to identify very rare variants that are associated with the disease. A vast majority of disease-causing variants are quite rare because most diseases are quite rare. Cholangiocarcinoma, being a rare cancer, is definitely one of those. And those genes and mutations are less obvious in small populations, which is why we're looking for large numbers of patients to be included in this study. So now I'm going to talk a little bit about what we do know about the genetics of cholangiocarcinoma, which is also going to point to what we don't know before going into the specifics of the project. So when we're thinking about genetics research, there are a couple of different reasons or goals that we have in performing this research. One is understanding risk. So in a patient who doesn't have a disease or cholangiocarcinoma, what is their risk of developing it over time? And this is really important, especially when it comes to someone who has a family member with a certain disease or cancer. It also helps us identify biomarkers. So some genetic variants may be more associated with severe disease as opposed to less severe disease, and they might be associated with different treatment responses or outcomes. So those are the biomarkers that might be relevant in your genetic sequencing results. And it also helps us develop targeted therapies. So there are certain variants that might respond well to certain medications, and so it's important to know what those are. And what we know about cholangiocarcinoma genetics right now is kind of that it's complicated. If you Google straight up just is cholangiocarcinoma inherited, you'll get a number of websites that say, no, it's not inherited. And this is one of the government sites, the National Library of Medicine, that says cholangiocarcinoma is not inherited. It says that most people with cholangiocarcinoma do not have a family history of the disease. Except, what does that most really mean? Because we do know that there are a number of people with cholangiocarcinoma with family history, which points to some sort of inherited mechanism. And so when you're looking at cancers in general, the National Cancer Institute says to up to 10% of all cancers can be caused by inherited genetic changes. And so when you're thinking most people don't have a family history, that's true of all cancers, but that 10% of the population, that's really important. And it's important to that person and their family members. And so there's been an increasing push over the past couple of years to really identify what that most is. Is it about this 10%? Is it less? Is it more? And how can we really address this gap in knowledge when it comes to cholangiocarcinoma genetics? So what we do know thus far is that a lot of these variants or single nucleotide polymorphisms that are found in cholangiocarcinoma are cancer-causing mutations. And these are things that were discovered with that candidate gene panel, where ahead of time the researcher identifies what genes they want to look at. Then they sequence those genes and look at those variants. And so these common cancer predisposing mutations, they're seen in a lot of different types of cancer and hereditary cancer syndrome. So an example of this would be Lynch syndrome or a BRCA mutation. So we are seeing that those sorts of hereditary cancer syndromes do predispose individuals to cholangiocarcinoma as well. It's also known that the genetic profile is likely different between intra and extrahepatic cholangiocarcinoma. And so this is pointing to an additionally emerging area of how similar or different are cholangiocarcinoma tumors based on where they present. And the small studies that have been performed have shown that the genetics of the patients who develop intra and extrahepatic cholangiocarcinoma likely are different. But all of these studies have been in really quite small numbers of patients, and they've been that candidate gene approach, like I talked about. So we're only testing for the things that we want to test for, which is great to confirm or deny that thought, but it doesn't give us new ideas. So this is one study that looked at cholangiocarcinoma germline genetics. So again, germline DNA is not the tumor DNA. This is from your blood. It's what you're born with, and it's what you can pass on to any children. So germline variants are variants that may not have been inherited from your parents, but they will be passed down to children. That bullet point, it's not worded well. So you may or may not have inherited it from your parents, but you would pass it to your children potentially. And so in this particular study, there were 136 cholangiocarcinoma patients. So again, relatively small numbers. That's quite small for a genetic study. And they looked at 84 cancer predisposing genes. Again, with that candidate gene panel approach, they pre-identified 84 genes that they wanted to look at. But what they found is that 15.7% of intrahepatic cholangiocarcinoma patients and 17% of extrahepatic cholangiocarcinoma patients did have a variant in one of these 84 genes that predisposed them to cancer. And so when we think back to that screenshot that I showed that said cholangiocarcinoma isn't inherited, we're seeing increasing evidence that that's certainly not entirely true. And in the boxes, those are the cholangiocarcinoma patients. If you're not familiar with protein names, which no one expects you to be, a lot of these are associated with multiple different cancer types. You see BRCA mutations. TP53 is a protein that's associated very strongly with cancer development. And so it's unsurprising to find these sorts of mutations in cholangiocarcinoma as well. This is a second study, quite similar. This is 146 patients, a different group of patients, again, a small number. They looked at 103 cancer predisposing genes, and 10% of those patients had a variant that was pathogenic or likely pathogenic. And so again, thinking cholangiocarcinoma is not inherited in this group of patients, we're again seeing that it's about 10% had germline genetic variants. And then additionally, about 50% had a variant of unknown significance. And this is how geneticists classify variants. And essentially, this variant of unknown significance is we can't confirm that it's disease causing, but we can't confirm that it's not either. So it's kind of in that middle category of we're not sure whether or not this variant is problematic. And so of this 51%, likely a lot of them are not disease causing, but some of them may be. So that 10.3% is kind of the conservative estimate, and it might be a little bit higher than that as well. So looking at the screenshots, cholangiocarcinoma is not inherited, but we just looked at some studies that showed that we are seeing germline genetic variants in cholangiocarcinoma patients. And if you're looking at all cancers, up to 10% of cancers might be caused by inherited genetic changes. And we're really seeing comparable data to that in cholangiocarcinoma. That's really important to know because it's associated with familial risk. A lot of people with cancer want to know whether or not their children have a chance of inheriting that same risk level. And germline genetic variants are also associated with earlier age of onset for cancer. So it's important to know whether or not that there's a variant associated with it for screening purposes. So going into the meat of what this project is, this is the International Consortium for the Genetics of Biliary Tract Cancers. This is a huge project that's led by individuals at several different institutions. So my boss, Dr. Lewis Roberts at Mayo Clinic, Dr. Manal Hassan at MD Anderson, Dr. Chris Amos at the University of New Mexico, and Dr. Catherine McGlynn at the U.S. National Cancer Institute. And so what we're leading is the first GWAS of cholangiocarcinoma. So our goal is to collect thousands of samples from cholangiocarcinoma patients and people without cholangiocarcinoma and compare the genomes between the two of them. So in the late 20-teens and early 2020s, in the past couple of years, we went through a discovery phase, which was essentially a pilot project to see whether or not this was worth pursuing. Are we going to get data that seems like it's robust and worth conducting a larger study on? So they collected about 2,600 cholangiocarcinoma cases and then controls as well from 24 different sites in North America and Europe. And this is a breakdown of the patient population that was used in this study. So relatively equal numbers of males and females. We're getting relatively equal numbers of both intrahepatic and extrahepatic cholangiocarcinoma. There's a kind of a small amount of PSC-associated cholangiocarcinoma, but we have reason to believe also that just like there is a different genetic profile between intrahepatic and extrahepatic cholangiocarcinoma, there's perhaps a different genetic profile between PSC-associated cholangiocarcinoma and non-PSC-associated cholangiocarcinoma. So it's important to have that distinction in our data analysis as well. So bringing us back to the diagram as a reminder, we're looking for variants that might be more or less frequent in cholangiocarcinoma patients. And I'm about to show you some of our preliminary data, which is going to look like this bottom panel here. And this is called a Manhattan plot. This is how data from GWAS studies is presented, and the individual dots are a specific genetic variant. And essentially, the higher up they are, the more confident we are that there is a true difference between the two groups that you're comparing. So if this was cholangiocarcinoma patients, there are some teal dots here and some black dots here that rise the highest. And so that's where we'd say we're quite confident that variants in that area are associated with cholangiocarcinoma. And we put two different lines on these charts. We put the line of statistical significance, which is what we use to formally declare we are quite confident that this variant is associated. Then there's also the line of suspicion, where we can't say with utmost confidence, but we have suspicion that this might be associated. So this is data from the discovery phase of the GWAS. So this is the 2,400 European samples, or samples from individuals of European descent. And we are seeing variants that are rising above that line of statistical significance in the red here. and then several more that are rising above that line of suspicion. And I want to draw your attention especially here to that big peak in chromosome 6. So this is what's called the HLA region of your gene, of your genome. And a side note on these HLA regions. So this is the part of your genome that codes for your immune response. And this is the most diverse part of the human genome, which makes sense evolutionarily because we want our immune systems to be quite varied so that if there is a new pandemic that's super severe, hopefully it wouldn't wipe out all of humankind because our immune systems are all built slightly differently so that we respond to different diseases. It's that abnormal variants in these HLA regions can predispose individuals to autoimmunity. And so autoimmunity can then be associated with cancer risk. And you can see from this paper here that this paper looked at a bunch of different types of cancers. I pulled out the data for biliary duct cancer specifically. So each square represents a different autoimmune disease. And the more red the square is, the more it's associated with bile duct cancer or cholangiocarcinoma. And so the ones with the A's are statistically significant, so we can say with confidence that they're associated. So this is to call attention to the fact that these HLA regions can predispose you to autoimmunity, which can then predispose you to cancer. And we also know that from cholangiocarcinoma epidemiology studies where primary sclerosing cholangitis, ulcerative colitis are very associated with cholangiocarcinoma development. And in our data specifically, we found several variants in those HLA regions that are associated with immune responses, autoimmune disease. For example, one of them was associated with lupus predisposition, and it's been associated with some other cancer types in the past, such as kidney and prostate cancer. So it's unsurprising, is the gist of what I'm trying to get here. It's unsurprising that we are seeing variants in these regions since we know that cancers, and bile duct cancer in specific, are associated with autoimmune diseases. When we break down the data between intrahepatic and extrahepatic cholangiocarcinoma, those dots tend to lower on the graph because now we're looking at a lot fewer cases. So this is only about half of the population. And the larger the study is, the more confident we are in the results, essentially. So in just the intrahepatic samples, we're not seeing things that rise above that line of statistical significance, but we are seeing things that rise above the line of suspicion. And same thing with extrahepatic samples. We're not seeing statistically significant things, but we are seeing suspicious things. And if you remember, this is the discovery phase of the project. And this data, even though there aren't things that are statistically significant in every group, is certainly saying that there are suspicious variants and that this warrants a larger study. So all of the data analysis for this discovery phase was done by Dr. Young-Hun Han, who is at the University of New Mexico. And what he found from this data set is that cholangiocarcinoma has an estimated heritability of 12%. So that is very similar to that 10% that we saw from the National Cancer Institute on overall cancer types. And so this is really working to change the paradigm of whether or not we think cholangiocarcinoma has that inherited component. We're seeing results that are very similar across studies that relate it to a similar heritability from other cancer types. We are seeing slight differences in that heritability between intrahepatic and extrahepatic cholangiocarcinoma, and then PSC-associated and non-PSC-associated cholangiocarcinoma. But we do need more resolution here in order to say with confidence that there are differences. The results of the study also identified some novel variants. So novel variants being variants that we don't really know much about, haven't previously been connected to cholangiocarcinoma. And this is a real strength of doing a GWAS study. as opposed to one of those candidate gene panels that previous studies have done, is that we wouldn't have thought to look for these novel variants had we pre-picked beforehand what we wanted to look at. And as I mentioned, a lot of the variants were in those HLA regions, which is unsurprising, but really good to know. And all of this data was very optimistic. We found statistically significant results, and we found a lot of suspicious results. So it really does warrant this expansion phase of the GWAS, which we're doing right now. So we were awarded a UL1 grant by the NIH, and that's one of the highest levels of grants that you can get. And it's a collaboration between all of those institutions that I mentioned earlier, the Mayo Clinic, University of New Mexico, MD Anderson, and the US National Cancer Institute. And our goal is to sequence 7,000 samples over 5 years, and we're just starting year 3 of that. So we have 3 more years left in this project, and our goal is to get to 7,000 samples. We're currently working with 82 sites from 31 different countries, and Cholangiocarcinoma Foundation is one of our big contributors to this project. And this will help us see with greater resolution which of those variants are strongly associated with cholangiocarcinoma. It'll likely help raise some of those dots from the line of suspicion to the line of statistical significance, so we can say with greater confidence that they are associated. And it'll also help us look at even rarer variants. So again, each of these variants is quite rare, cholangiocarcinoma is quite rare, and we really need these large sample sizes in order to be able to identify rare variants associated with CCA. A big part of this project is also an emphasis on global genetic diversity and increasing representation. So if you remember back to the discovery phase of the project, all of the samples were from patients of European descent. And a large part of the reason that that was the case was because American and European institutions who store samples are largely seeing patients of European descent. So it was an availability bias. So in this expansion phase, we're really working on having a broader scope in the types of patients and control samples that we're collecting. And this is really important because certain genetic variants are more common in certain global regions. So if you think of the evolution of humanity, if millions of years ago, one individual in a certain region developed a certain genetic variant and then passed it down to part of their progeny, you're likely only going to see that variant around that certain region if there's minimal migration. And so in studying only patients of European descent, we're missing out on a lot of global genetic diversity. In fact, there are studies that show that if you were to classify all humans into 6 different groups genetically, 5 of those groups would be in Africa. And so there are 5 African groups out of the 6, and then 1 of everyone else out of the 6. And so there's huge global genetic diversity that most genetics research has missed in the past, because a lot of that development is happening in North America and Europe. Cholangiocarcinoma, with the caveat that there is a lot of Southeast Asian research on it because of the prevalence there. And so one of the ways that we are approaching this is that the way that we're sequencing it is something called a global diversity array. This array looks at 4,800 genes and over 1.8 million variants. And it looks at variants that are not only just prevalent in overwhelmingly white populations, but it took genomes, when they built this, they took genomes from across the globe to try to identify the variants that might be relevant to certain populations as opposed to others. And I'm pulling you back to that map I showed you at the beginning. I told you to notice which areas of the world were grayed out. And this is a current map of countries that we're working with as part of our consortium. So you can see we have the United States, Canada, and a lot of Europe, but we're also working with many sites across South America and Africa, especially in order to increase the global genetic diversity that's represented in this study. And like I said, Cholangiocarcinoma Foundation is one of our major contributors. And so there are ways that you too can participate in this study. This study, like I said, it's a 5-year project. So if you are contributing a sample, it won't be sequenced for about another 3 years. And because of the way that we're sequencing it, results aren't returned to patients. So that's important to know when you are thinking about whether or not you want to participate. This isn't a study that will be able to inform any clinical decision making for you, since we're looking for those large numbers in order to see differences. So this is really a pay it forward type of study. So hopefully the results of this study will inform future clinical care for future patients. And there are 2 different ways that you can participate depending on whether or not you're going to be attending the conference in 2 weeks. So for people who are not attending the conference in Salt Lake City, you can use this QR code here. It'll take you to the CCF website, and they have a form there that you can fill out. You'll submit your contact information. Dr. Muhammad Ali at Mayo will send you a blood kit. You bring it to a clinic of your choice. Costs are covered. You donate a small blood sample, and then that will be shipped back to the Mayo Clinic for sequencing. And like I said, we need both cholangiocarcinoma patients and people without cholangiocarcinoma. And so this is open to both CCA patients and then as well as caregivers, families, friends, anyone who might also be interested in participating. And then for those who are attending the Cholangiocarcinoma Foundation conference, we are actually going to have a team there in person to do blood draws. And so this QR code will take you to a sign up demographic form to get you consented into the study. And then there'll be a room in the main conference area. There'll be signs, and we'll have phlebotomists there that can take your blood sample at the conference in order for you to participate. This is our 3rd year doing it, and it's been quite successful in the past. We really appreciate the willingness of CCF, people involved in CCF, in order to contribute to this research project. And we're excited to see hopefully a number of you in person as well. So this is like a non-extensive list of everyone who is helping to contribute to this project. But it's a huge lift from a large number of people, and we really appreciate both the investigators and the patients who are involved in this study. And I'm happy to take any questions. So I can see the chat from here. Let me see if I can see
I
and
can read the couple that have come through if you'd like, Caitlin.
Okay.
Thank you, first of all, for your presentation. It's very informative, as always. Someone asked, like a clarifying question, variants may not be inherited, but you can still pass it on. Does that mean that somewhere a CCA patient's DNA mutated and once is, then it may be passed on?
Yeah, that's a great question. And so there are 2 different types of germline genetic variants. There's inherited variants, and then there's what's called de novo variants, and that means like a new variant. And so these are all early in embryonic development. So when your cells are first dividing in the womb, an early variant there can cause a de novo mutation. And those are the kinds that you didn't inherit from your parents, but you can pass on to your child. I hope that answers that.
Then the other question, there are several patients in our community with an immediate family member with CCA. Has there been any studies on patients with a family member with CCA?
Yeah, and so that's also a good question. That's what this study is really also hoping to look at. So in the demographics form that you fill out, you can denote whether or not you have a family member with cholangiocarcinoma, and that will be taken into account in the analysis.
Does 12% heritability propose that I have a 12% chance of passing down a disposition to CCA?
Mm-hmm. So heritability, I should have explained this. I should have explained what this term means. It's definitely a piece of jargon. So heritability is the percent of risk that can be contributed to genetics. So it's not that a single individual has a 12% risk of passing it on. It's that out of everyone with cholangiocarcinoma, we estimate that about 12% of the risk has to do with genetics. It's kind of a technical difference, but it's about a population level thing as opposed to an individual patient level thing. So not quite.
Okay, perfect. So this study is not meant to have results, and there's no way that someone would get results, correct?
Correct, yeah.
Okay, there's been a couple of questions, and you may or may not be able to answer this. If you have any recommendations on screening for family members, on how to be proactive.
Yeah, so I would definitely encourage you to talk, or you or whoever is the patient, to talk with their oncologist about screening and what they recommend. I think that bringing up concern about increasing information on the heritability of cholangiocarcinoma is something that a lot of physicians would be willing to talk to you about. And they can recommend from there what sort of screening they think is appropriate.
Okay. Are we able to see the list of questionable CCA SNPs that have already been identified?
Yeah, good question. So all of the data that I showed right now is in its last stages of being put together before we publish it. And so when that is published, hopefully soon, it's been a process. We will definitely be sharing those results with the Cholangiocarcinoma Foundation that I think can then be distributed um, to all of you.
Okay, perfect. I know that you said that you're looking in the study for patients, more diverse genetic populations. If we have, the foundation has a lot of interactions with people in other countries, is there any specific sites that we can direct them to depending on the country that they're from?
There are, and so you or whoever is handling this should feel free to email me about any specific country. Depending on the country, we may also be able to ship a blood draw kit directly to that patient.
Okay, perfect. That's good to know. I don't see any other questions at the moment. If anyone else has another question,
I see one,
please. Okay.
if a parent with CCA has, if a parent has CCA, what the recommendations would be. Again, that would be talking to their care provider or your care provider about whether or not they think genetic screening is appropriate in that case.
Um, so this is a question about understanding whether or not your current doctor should be able to recommend screening procedures to other family members. Is that?
Yeah, they should be able to, and feel free to bring up your concerns about genetic risk. That's really important in those lines of communication.
Okay, perfect. And then for those who are asking, this is being recorded and the recording will be emailed to you in about 24 hours. So look in your email for that link.
I see there's a question on will the cuts at the NIH impact this study? That is something that we are currently waiting to find out. So we just submitted our progress report for year 2. We're waiting for our funds for year 3 to come out. So far we don't have reason to think that it will be drastically cut, but NIH funding overall has been cut. And we are waiting to see how much this will impact this study.
Okay. I
I'm
don't
seeing
think you can
also
answer
a
this
question
question.
about middle-aged twins who had
Uh-huh.
cholangiocarcinoma were seen at MD Anderson. I'm not aware of this specific case. I can talk to our colleagues at MD Anderson and see if they are.
If anyone else has any questions, please feel free to put your question into the chat or the Q&A. Otherwise, thank you, Caitlin, for this update. It's been really helpful. And yes, someone who donated blood a couple of years ago to Mayo Clinic for the CCA study, this is the same study.
Yeah, good question.
Okay, somebody has a question, and I think that they're typing it, so I will wait. If international patients are coming to the conference, can they participate in the study?
Yes, absolutely.
Okay. Do you encourage family members to participate in this study?
Yeah, we absolutely do. Again, all comers are welcome. We are particularly interested in that familial link to see how a non-affected family member's DNA may or may not be different from their family member with cholangiocarcinoma. But and anyone can participate, yeah.
Okay, perfect.
Repeating what I said about the twins at MD Anderson, there was a comment from Matt Reddy about a case of middle-aged twins who both had cholangiocarcinoma were seen at MD Anderson. I was just saying that I don't know of that particular case, but I can talk to some of our colleagues at MD Anderson about it. And Leanne, that's a great question, and that's something that our analysis team takes into account when they're looking at the data.
So the question that was asked was if whether or not the information, if family members eventually go on to develop CCA, wouldn't that skew the results? And that's what Caitlin was responding to.
Yeah, that's the kind of thing where we're using the specialists of specialists for bioinformatics for those really nuanced analyses.
If someone has twins, one who has CCA, I don't know if you can answer this, but what's the chance the other twin will develop it?
It is it's pretty impossible to say based on an individual level. So there are a ton of different risk factors for cholangiocarcinoma, and not all of them are genetic. Um, and so it really depends on the cause of the first twin's cholangiocarcinoma. And so that would be a situation in which talking to the provider about genetic screening would be very useful.
Perfect, thank you. I don't see any other questions coming through. Thank you again, Caitlin, for this presentation and the update. We appreciate it. Looking forward to more information as the research continues.
Yeah, thank you all so much for joining. And
Thanks,
hopefully I'll
everyone.
see a number of you in Salt Lake City next month.
Yes, definitely. Thank you, everyone.